This is a bit of biology with Mr. Rock, and today we are going to be doing the Unit 2 review on biochemistry. This video is going to explain the major ideas behind Unit 2, which we did, and that was biochemistry, but I also do briefly just want to insert some stuff on scientific method. Scientific method is an elaborate process of how we go about learning things in science. However, the three things that I just want you to understand for right now is what your variables are. And in every given experiment, there are going to be three types of variables. If we're going to talk about me, Mr. Rock, growing plants for an experiment, and I'm going to do an experiment where I change the light, the type of light or the amount of light, and I'm going to measure how tall the plant gets. That's a pretty basic experiment, but I want to do that in my class to learn how amount of light affects plant growth. Your independent variable is going to be the thing that you specifically change or manipulate in an experiment. So if I give my example of plant growth, my independent variable is going to be the changing amount of light, because that is something that I am specifically changing as an experimenter. Your dependent variable can best be remembered by the variable that you measure. In my experiment, you can ask your question, what are you measuring? I will ask myself that question. What am I measuring? I am measuring plant growth via measuring the stem. So I'm measuring the stem of the plant, and that is my dependent variable. So the dependent variable is what you measure. I'm measuring stem growth. Finally, your controls in the experiment, your control variables are things that you want to keep the same. In an experiment, you want to have lots and lots of controls. If I'm doing this experiment, I only want to see how the amount of light influences plant growth. So I do not want to see how different water sources, different temperatures, different fertilizers, all of those things I want to keep the same, because if I change those as well, I don't know what's affecting the plant growth. So in my experiment, controls would be keeping the amount of water for each plant the same. It would be using the same type of soil, putting the plants in the same kind of room with the same temperature. So all of these are controls. You want many control variables in an experiment. Okay, so the true purpose of this video is to review biochemistry, and we had two main topics in biochemistry. One, we had macromolecules, and two, we had enzymes. So there are four macromolecules: carbohydrates, lipids, proteins, and nucleic acids. And with that, there are four main things that I want you to know with each one. So the function, the monomer, polymer, and an example. Carbohydrates, let's start with them, and I'm actually going to jump down to the example. With these macromolecules, you know about them because they're on the back of your food labels. Macromolecules are the foods that we eat and how we use them in biology. So an example of a carb is bread. Also rice, sugars, those are carbs. What is the function of carbohydrates? The main one is energy. Okay, we use carbs for energy. That's why you carb load before a big run. But then also in plants, they use structure. So it's structure in plants, and they use this polymer polysaccharide called cellulose. So it's structure in plants and energy for all organisms. The monomer, jumping back to that, monomers are little tiny Lego pieces. So monomer is just one Lego piece, and you put a bunch of monomers together to make a gigantic Lego structure, like the Millennium Falcon. That's what I built as a kid. So your monomers are your little Lego pieces. The polymer is the final end product. So the monomers for carbohydrates is a monosaccharide, which is a bigger word, but an example would be glucose. And you saw glucose before because you saw that in the energy flow unit where we need glucose and oxygen to make ATP. So definitely need glucose for that energy. Polymer, the polymer for carbs is a polysaccharide. There's a lot of different types of complex carbohydrate molecules, but an example that you would know here is starch. Okay, we're going to move on to lipids. Lipids, a good example of that would be butter, oil, fatty things, grease. Those have a lot of fat in them. Also like some types of meats do too. The purpose of lipids have a couple more functions. So one, it's long-term energy storage. So you can use fat for energy, but it's like a backup storage, energy source. You could use it for insulation to stay warm. But then the big thing here that I want you to remember from the cells unit is it makes up your cell membranes. The cell membrane is made up of a phospholipid bilayer. Our monomers, or little tiny Lego piece, is going to be a fatty acids, and they're going to work together, build together to make the polymer, which is a triglyceride. Moving on to our proteins and nucleic acids. Proteins serve a lot of functions in biology. The big one that I want you to remember is from DNA. We use DNA to make RNA, RNA to make proteins. Proteins are structure in life, and we call them the building blocks of life. So structure, building blocks of life, that's the function of proteins. Also, they do a lot of other things like make up the cell membrane. So they use protein channels and protein receptors and things like that. And then also, finally coming in this video, they make up enzymes. Proteins make up enzymes. The monomer is amino acid. Once again, think back to translation and DNA. You saw an amino acid chart, which you used mRNA to figure out what, like alanine, leucine, all of those things. So amino acids are the small little building blocks, and they come together to make a polypeptide chain, which is our protein. And an example of protein that you eat every single day is probably meat. Finally, nucleic acids. Big thing here is to store genetic information. That's why we need nucleic acids. Just jumping down a little bit to the polymer. The polymer, DNA, deoxyribonucleic acid. RNA, ribonucleic acid. So anything that you eat that has DNA would have nucleic acids. And finally, the last thing I want to say is the nucleotide is just the small Lego piece for making the huge double helix DNA. So the nucleotide is just one building block, that's the monomer, and it makes the huge double-stranded double helix DNA. Finally, what are enzymes? Enzymes are biological catalysts that speed up chemical reactions. If you say from that definition, well, Mr. Rock, what is a catalyst? I don't remember that. Catalysts are things in chemistry that speed up chemical reactions. So, simply put, enzymes speed up chemical reactions. So we use enzymes a lot in our digestive system to break down huge polymers, and we want to break them down into monomers. So you eat starch and you want to break that starch down to glucose. We use enzymes to do that. Enzymes are, so a couple characteristics about them, they are proteins. They are also specific, so they work like a lock and key. The enzyme is the lock, the substrate, which is what the enzyme works on, is the key. So only protease is going to break down protein. Lipase is going to break down lipids. So lipids are only broken down by the lipase enzyme. Another thing to know about them, they are reusable. As you just heard with my examples, they usually end in ase, and they are also affected by temperature and pH. So if you change the temperature, your body temperature too much, that can destroy enzyme activity. It messes up the lock, and if the pH gets changed too much, you can also destroy the lock. So that affects the enzyme activity, and you would have remembered this by breaking the toothpicks with your hands. I don't know if you remember that, lab, but you broke the toothpicks with your hands. If you put your hand in a bucket of, ice, you're going to change the enzyme activity. Final points would be just right here. 90% of the time, enzymes end in ase, but that's not always true. Sometimes you'll see an enzyme that doesn't end in ase, so don't freak out if you see that. And then finally, we do talk about enzymes a lot in digestive system and breaking down polymers into monomers. But enzymes can also synthesize and make new things. An example of that is like DNA. Helicase, polymerase, all of those words that you saw during DNA replication, those are all enzymes, and they're making something new. They're building something. So enzymes can break stuff down, but then they can also speed up the process of synthesizing or creating something. This has been a bit of biology with Mr. Rock. I'm signing off.